Hi students, a very happy evening to everyone present here. I welcome you all to the day 3 of refresher course. So today we have with us Dr. Ajith Madhavan, who's going to give you an idea about the biology topic. So he'll be covering packaging of DNA helix, translation, transcription, and genetic code. So be ready for this session. And if you have any queries, please make use of the chat box. Please type your queries there. We'll be addressing it towards the end of the session. So I welcome you, sir, towards the session.
Thank you very much, ma'am. Thank you. Shall I start sharing the screen, ma'am?
Yes, sir, you can start sharing the screen.
Yeah. Shall I start, ma'am?
Yes, sir.
Okay, thank you so much, ma'am. Thank you so much for arranging such a platform, which I thought is very important for dissemination of biological information. We get to teach only the college students, but it would be interesting to talk to school students as well. Thank you so much for the opportunity. So today, we'll
sir,
have an interesting discussion.
Sir, kindly hold on for a minute. You have not shared the screen.
Okay, ma'am. Sorry.
Yeah.
Now it's shared, is it not, ma'am?
Yes, yes. No, I think it's like moving towards the end. You can take the first slide, I think. Yes, yes.
Okay. Thanks, ma'am.
Yeah.
Sorry for that. Today we'll have an interesting discussion, not because the discussion is going to be interesting, rather the topic itself is interesting. Let's talk about the packaging of, as ma'am mentioned, packaging of DNA helix, then the transcription process, the genetic code and translation. So let's right away move to the packaging of the DNA molecule. So before going into the packaging of DNA molecule, what we need to understand is, if you take a eukaryotic cell, for example, which is really small, around 5 to 100 nanometer, 5 to 100 micrometer in size, and within which you have a spool of DNA, which is the blueprint of the cell. The information required for the structure and the function of the cell is present inside the inside the cell as in the form of a DNA molecule, a double-stranded DNA molecule. So they are basically packed in separate units, especially in eukaryotes, called as chromosomes. But let's imagine all these DNA are unspooled. They are connected end to end. In fact, it will extend to a length of 2 meters. So we are looking at 2 meters of DNA being packed inside a small space of, for example, 100 micrometer. But the problem is, it can't be very randomly packed inside the cell, because the information present in the DNA molecule has to be retrieved. So it shouldn't be entangled. It has to be neatly packed, orderly packed and placed inside the cell. In a eukaryotic cell, inside a nucleus. In a prokaryotic cell, in a particular nucleoplasm, which is attached to the cell wall and so on. So how it is packed? How it is packed is the question. So in the case of eukaryotic cell, imagine you have a single stretch of DNA molecule. Each chromosome, I told you, is made up of roughly around 5 centimeters of the DNA molecule. They are condensed or in an orderly fashion into a 60 to 70 millimeter chromosome. How it is done? It is done at different levels. So the naked DNA is actually wound around a protein cylinder. The protein cylinder is an octameric protein. Octamer meaning it is made up of 8 proteins, and the protein is called as an histone. Around the histone, the first level of condensation happens. So how much is spooled around the histone? It is between 140 base pairs to 150 base pairs. It is spooled around. So you have cylinder-like particles and the DNA molecule is spooled around it. Like we have threads which is spooled into a bundle. So that's how it is carried out. So that is the first level. Around 140 bases to 150 bases is spooled around it. So you can see in this picture, especially this one, you can see this is a cylinder around which the DNA molecule is spooled. And in order to stabilize the spooling, you have another glue protein, which is called as a linker histone, which is bound and the spooling is stabilized. And the DNA plus the histone, it is called as nucleosome, some meaning body. So the first level is the naked DNA spooled around the histones, octameric histones. And after that, so this histone plus the DNA molecule, it is around 10 nanometer fibers. The DNA, naked DNA is 2 nanometer. When it is spooled around the histone, it becomes 10 nanometer fibers. And the 10 nanometer fiber is again spooled, again wound into structures like this. Now you can see it is wound like this as a phone cord. So it is wound in the form of a solenoid or it is wound in the form of a helices. So this is the second level. So the naked DNA is wound around the octameric histone, which is called as a chromatin, and the chromatin, which is now little thicker, 2 nanometer becoming 10 nanometer, and the 10 nanometer is wound into a helical structure to form a 30 nanometer fiber. The 30 nanometer fiber, again, by a complex winding, forms a chromosome. So you can see a 5 centimeter stretch of DNA is wound into around a 50 to 70 nanometer particle. So this diagram again sort of helps you visualize what I've been saying. So don't need to worry about this picture. So this is the edge of a cell from which the DNA has been removed. The DNA is oozed out. So the hazy region that you see is the DNA. Now what happens? The DNA is now wound into a 10 nanometer fiber. Then it is again wound into a 30 nanometer fiber, then into a chromosome. So if you are not convinced with this, again, look at this picture. So the best way to sort of register this image is looking at the schematics. So you can see the chromatin fiber. So you can see how much of DNA is present inside the cell. Huge amount of DNA. Then after that, I told you, see here, the 10 nanometer fiber. So the DNA is being spooled around the histones, and it is again spooled into a 30 nanometer fiber, and the 30 nanometer fiber again spooled into a chromosome. So this helps you to understand the condensation of the DNA molecule. But it is also interesting to imagine, you may ask a question, okay, the DNA molecule is wound around the protein, but the information resident in this DNA molecule has to be deciphered, has to be read. So the protein molecule might become a hindrance for that. So there are different ways by which the cells deal with this problem. They temporarily unwind the DNA, then read them and wind it back. And the histone protein that I'm talking about is a very active place. It not only carries out this mundane activity of condensing the DNA molecule, the expression of the DNA molecule itself is associated with histones. I'm not dwelling deep into it. If time permits, we can do it in a different discussion altogether. The modification is called as epigenetic modification. Just remember that terminology, that is enough. Having said that, let me be a little quick so that I can cover the topics that we have already promised to cover. So before going into the deciphering of information from the DNA molecule, the DNA, you know that it's a linear molecule. It is double-stranded. Like in a staircase, it is helically wound. So the two strands are helically wound. And the DNA molecule are made up of nitrogenous bases. They are nitrogen-containing chemical moieties. The nitrogenous bases are connected to phosphate and sugar to form nucleotides. It comes in 4 varieties: adenine, thymine, guanine, and cytosine. Okay, so this is the information. The sequence is the information that is present in the DNA. The language with which it is written, the information is written in the DNA, is by these 4 letters. The nucleotide is the letters that we are talking about, the 4 letters. If English is written in 24 letters, you have 4 letters writing up the book of life itself. Okay, that is not the important point. The point is, fine, the information is present in the DNA molecule. How it is deciphered? You need to read it. Otherwise, what is the use? Information needs to be realized into something. So what really happens is, the information is deciphered in 2 stages. So the information present in the DNA is converted to a sort of a similar information, which is called as transcription. So what is transcription? For example, you can write a particular word spoke in Malayalam into English. So you can write a Malayalam word in English. The meaning doesn't change. The sound of the word doesn't change, but it is written in a slightly different language. So the process itself is called as transcription. So in this case, the DNA information is written in an RNA information. The DNA and the RNA are in fact sort of close cousins, chemical cousins, I would call. They only have a difference of having an extra OH group in the case of RNA molecule, or in the case of nucleotide that goes into the making of the RNA molecule, at the second carbon of the sugar, you have an extra OH. In the case of DNA, that OH is missing, hence deoxy. Deoxyribonucleic acid. This is ribonucleic acid, RNA. The DNA is written into RNA molecule. The process is called as transcription. After that, the RNA molecule is converted to a different language altogether. So the Sounding of the word is different, but the meaning is the same. It is meaningful. I shouldn't say the meaning is the same. It is meaningful. It's meaningfully written. So the information written in 4 letters of nucleotide is converted to another language and information that is written in 20 letters. The letters are called as amino acids, and the sentence and the words, they are called as proteins. Now you know the flow of information from DNA to RNA by the process of transcription to proteins by the process of translation. So this flow of information was in fact proposed by none other than Francis Crick, the co-discoverer of the double helical structure of the DNA molecule. But it's very interesting whether, if you ask me a question, whether the flow of information is unidirectional, whether it always moves from DNA to RNA to that of protein. So far, total reversal of the information is not seen in the biological system so far, but partial reversal is seen. Meaning, the information present in the RNA molecule is the information present in the RNA molecule in certain instances is converted to DNA molecule, especially in certain viruses. For example, the one that causes HIV. So they are called as retroviruses. No need to worry about that. We have to be a little quicker. Okay. Now, what is transcription? So transcription, I told you, it's the conversion of the DNA information into RNA information is called as transcription. So listen very quickly. I've already told you this is a double-stranded DNA. So from the double-stranded DNA, an RNA molecule have to be synthesized. So looking at this diagram, it gives you some important information. One, the DNA molecule is double-stranded. It is locally opened up so that the 2 strands are separate. On one of the strand, only on one of the strand, only from one of the strand, an RNA molecule is synthesized. That strand is called as a sense strand. The other strand is called as an antisense strand. Okay, so what do we mean by that? Look at this particular diagram. So see here, the DNA is a beautiful structure. It is like a zip. You can imagine it like a zip. You move the rider so that the zip opens up. You have 2 parts of the zip. Now, both these strands of a DNA molecule can serve as a mold, can serve as a template for the synthesis of an RNA molecule. How is the question? So listen very carefully. The DNA opens up. Then to the nucleotide, you can add 1 nucleotide at a time. So how it is added? Because in biology, especially in the DNA and the RNA molecule, the bases show complementarity. What is complementarity? For example, if you take a pen, you have a cap that is complementary to the pen. Probably you can't use the cap to any other pen. It is exactly fitting. Likewise, the adenine fits perfectly with thymine, and the guanine fits perfectly with cytosine. So now you know, when you open up the DNA, you have a strand. The strand has sequences. Sequences are what is information I told you. If you have an adenine and a U will be added to it. So what is the information? Look at the second picture in this diagram. Listen very carefully. So the DNA template I told you, whenever there is an adenine, a uracil will be added. The second difference of an RNA molecule. The first difference I told you, at the second carbon position of the ribose sugar, an OH is missing in the case of DNA. You have an excess OH at the RNA molecule. Another difference is you don't have thymine here. Instead of thymine, you have a modified version, which is called as uracil. That is the second difference. So in the DNA molecule, in the template, the opened up template, if adenine is present, a uracil is added. If thymine is present, an adenine is added. If guanine is present, a cytosine is added. If cytosine is present, a guanine is added. Now you can see the information that is present in the DNA molecule is rewritten into a similar molecule, which is the RNA molecule. Okay. Now, very quickly, I'm not little satisfied with the description. I want to just okay. So what I'm trying to say is, listen very carefully. This is the DNA molecule. This is the DNA molecule. Imagine you have an A, T, G, C, A, imagine. An RNA molecule will be synthesized by adding a uracil here, then an adenine here, and as it is added, they are connected. The connection in this case, we call it as a phosphodiester bond. It happens, you all people know that, between 3' OH and a 5' phosphate. Then a C will be added, it will be connected. A G will be added, it will be connected. A U will be added, it will be connected. Now you can see this is the RNA molecule and this is the DNA molecule. This is how it is synthesized. Hope you understood. If you have any doubts, save it as a question. We'll try to answer that. Now, another beautiful question that you can ask is, fine, I told you, complementary bases has to be added. The bases are slightly different. One is called as RNA, one is called as ribonucleic acid, the other is called as deoxyribonucleic acid. Fine. Then it has to be connected with a phosphodiester bond. So this amazing act should be carried out by somebody. It won't happen by itself. So who carries it out is the question. You have a beautiful molecular machinery, which is called as RNA polymerase. Technically, it is called as DNA-dependent RNA polymerase. You know why? Because the RNA molecule is synthesized from a DNA molecule. Hence, it is called as DNA-dependent RNA polymerase. So they come in different varieties with regard to prokaryotes and eukaryotes. So in the case of bacteria, it is made up of a beta-dash protein, beta, alpha, and so on. There are different types. Likewise, in the case of archaeal protein, you have different types. Likewise, in the case of eukaryotes, again, you have RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and so on. So they are called as polymerase 1, polymerase 2, polymerase 3, and so on. So usually polymerase 3 is the active polymerase that synthesizes the mRNA molecule. So the polymerase, in fact, has a crab claw shaped structure. People call it as a crab claw shaped structure. So you can see a crab claw shaped structure with two pincers of the claw and leaving a space in between. This is how it is. The enzyme that synthesizes the RNA molecule is called as DNA-dependent RNA polymerase. Now listen very carefully. I'm very quickly going into the process of okay. So see here, the transcription can be divided into 3 phases: initiation, elongation, and termination. It is called as initiation, elongation, and termination. 3 phases of transcription. Okay. So the question is, let me ask you another question, probably that demands an answer. You may ask a question, fine. The information present in the RNA molecule and the DNA molecule are quite the same. Why does the cell take all the pains in order to create an alternative structure, expending energy and material? The biological system does nothing without any purpose, because doing something meaning expending energy and material. So what is the use of transcription? The use of transcription is to control the retrieval of information from the DNA molecule. So you have a CD disk. You always do not want to read the information from one end to the other. You want at times read only certain information. So a selective retrieval of information is required. So that is exactly for the purpose you have a process called as transcription. Transcription will happen only in certain sections of the DNA molecule. The whole of the DNA molecule is not transcribed. Only certain genes will be transcribed at certain point in time. That's exactly the reason why your skin cell is different from the nephrons, the kidney cell, which is different from the neurons, the brain cell, which is different from the epithelial cells, which is different from the sperm cells, the egg cells, and so on. The information present in all these cells are same. 23 pairs of chromosome. But why do the cells look different? Because of the process of regulation. One of the effective ways in which the cell can regulate the information is at the transcriptional level. So in neuronal cells, only certain genes will be transcribed. Hence, it looks like a neuronal cell. In nephrons, only certain sets of genes will be transcribed. Hence, look like a nephron. In a pancreatic cells, the profile is different, and so on. So what I'm trying to say is, the transcription is a local mechanism. It is not global. Local in the sense that the whole of the information present in the DNA molecule is not transcribed. Only certain local regions, depending on the cell, is transcribed. So having said that, when I say only local sections of a DNA molecule is transcribed, I also imply that there should be a start point and there should be a stop point. Only then certain segments will be transcribed. There should be a start point and there should be a stop point. So for synthesis of an RNA molecule, from a start point to a stop point happens in 3 phases: initiation, elongation, and termination. Initiation, elongation, and termination. Okay, so the polymerase that I told you identifies certain segment of the DNA molecule. That segment of the DNA molecule is called as a promoter because it promotes transcription. You have a segment which is called as a promoter. It goes and binds to that segment. Now the polymerase has identified the start point. Once it has identified the start point, the binding is called as a closed complex. Why? Because the double-stranded DNA molecule has not opened up. Hence, since it is closed, you call it as a closed complex. Then what happens? Something happens to the polymerase. It applies some stress on the DNA molecule. The DNA opens up. The process is called as an open complex. The process is called as an open complex. Once the polymerase binds to the promoter, moving from closed complex to open complex, when it is ready to be transcribed, because the DNA is opened up, the 2 templates are opened up. On one of the template, synthesis is going to happen. Sorry. When the closed complex, the first step is called as initiation. So the first step is called as initiation. After that, the polymerase starts moving from the promoter to the stop point or the termination site. As it is moving, it adds 1 nucleotide at a time and connects them together by phosphodiester bond. So the process is called as elongation. Once it has reached a termination site, the polymerase is removed from the template. The polymerase is removed from the template and the RNA molecule is released. Now let me walk you through the process again. You have a DNA molecule. There is a small section which indicates that this is the point where transcription has to happen. I told you indicates that. So you have to have some other process to indicate when it is time for that region to be transcribed. Don't need to worry about that. So the polymerase goes and binds to the promoter. After binding to the promoter, closed complex, open complex. So sorry. So closed complex, from closed complex to open complex, from open complex, elongation phase, it moves from the promoter to the stop codon, promoter to the termination site. So sorry, it moves, reaches the termination site. The polymerase removes from the template and the RNA molecule is released. So this is what transcription is all about. It happens in 3 phases: initiation, elongation, and termination. Let me give you some details, relevant details, and we can stop there with regard to transcription. So the question is, listen very carefully. Don't need to too much worry about all these things. Excellent it is. In the future classes, you can actually, in your career, you can dwell deep into these aspects. Really, really interesting. Now the question is, I told you, I'm zooming in on the promoter. The promoter is the site where the polymerase goes and binds. What is the architecture? What is the structure of that promoter? So the promoter is a nucleotide sequence, there is no doubt. It's a DNA molecule. So the DNA molecule listen very carefully, has 2 regions. These 2 violet colored boxes that you see are the 2 regions. It is called as the -35 and -10 region. I have to give you some background information about this. So listen very carefully. So you have a DNA molecule and it has sequences A, T, G, C, A, T, C, T, A, and so on. Imagine it is at this T, the transcription is going to happen. So this nucleotide is designated as +1. So towards the right-hand region, it is called as downstream. Towards the left-hand side, it is called as upstream. So it is like a river. From the catchment area, the river flows down. So the catchment area is called as an upstream, and the valley into which it flows is called as a downstream. So see here, +1. Towards the downstream, it is 2, 3, 4, 5, etc. That's how you number it. Towards the left, it is -1, -2, -3, and so on. Now remember this numbering. Let's go to the picture. So this -35 meaning there are 6 nucleotide around minus 35 nucleotide, which is very, very important. You have a minus 10 region, which is very, very important. How many nucleotides are there? 6 nucleotides. Between this minus 35, 6 nucleotide and minus 10, 6 nucleotide, you have to have a distance of 17 to 19 base pairs. Because it's a double-stranded DNA, you call it as base pairs. Because for each nucleotide of a strand, a complementary nucleotide is present in the other strand. So they pair together, base pair. So this is the architecture. So don't worry about the other 2 pictures. Those are variation of this architecture. Don't need to worry about that. Now, why did I say that? So the polymerase recognize this pattern. You have to have a minus 10 region, a minus 35 region. In certain instances, you can see an UP element. UP meaning upstream element, nothing to worry. You know what is upstream? Upstream element. So the polymerase, how does it recognize a promoter? Using this site. The polymerase has an important region which is called as a sigma factor. It is called as a sigma factor. So imagine you have a polymerase which is in the shape of a crab claw. Then in the claw, you have a factor which is called a sigma factor. It is through the sigma factor, it goes and identifies the promoter. The sigma factor has different region, sigma 1, sigma 2, sigma 3, sigma 4, and so on. So you know, sigma 2 and sigma 4 are the 2 that recognizes the minus 35 and the minus 10 region. Now imagine in your mind, you have a plug point. You have a plug. The plug and the point has to have correctly registering components. Then you plug it correctly. Likewise, the sigma factor has 2 regions, sigma 4 and sigma 2, by which it correctly identifies minus 35 and minus 10 region. The polymerase this is how it identifies the promoter. So just the right information. Okay. So let's not worry about this. This is little complex. No need to worry. Now, the polymerase, they move one nucleotide after another. The polymerase, 1 minute. The polymerase moves one nucleotide after another and it reaches a stop site. It reaches a stop site. So now the question is, how does it stop? Now I'll give you an analogy. Never in biology take an analogy too far. Whenever it serves the purpose of explaining what is being discussed, that is enough. Don't take that analogy too far. It would be misleading. So listen very carefully. This is the RNA molecule and this is the DNA that is opened up. The bubble opens up and reaches the point where it has to stop. Where did it start? From the promoter. So how does it stop is the question. There are 2 ways by stopping. One is called as row dependent. Another is called as row independent. Row meaning RHO. So the row is a protein basically. One of the processes is dependent on this protein, hence it is called as Rho dependent. One of the mechanism is not dependent on this protein, hence it is called as Rho independent. Now I'm going to very quickly discuss about what is Rho independent mechanism of stopping transcription. What is stopping transcription? The newly synthesized RNA molecule has to come out and free, ready for translation and so on. And the bubble has to close, done. So I'm giving you a small analogy. What is that analogy? Imagine you have a pole. To the pole, a thread is tied, a cord is tied. It is little loosely tied. And you want to release the rope from the pole. What do? You make a tug. You tug it. You apply stress so that the rope comes out of the pole. Like similarly, in the case of the transcription process, whenever it reaches a stop point or the termination point, I told you, you can even take the analogy next step. It is loosely tied. In this case, you can see the termination point is marked by AU base pair. So you can see a series of U here. It is AU base pair. What is so special about AU? I told you loose tying. A and U are connected by 2 bonds. And G and C are connected by triple bonds. So the G and C are stronger in binding, and the A and U are less stronger in binding. I told you the rope is tied to the pole loosely, so the tugging can remove it. Now the question is, who tugs it? Who actually stretches it? Listen very carefully. Beautiful mechanism it is. So what happens is, the RNA molecule, when it reaches the termination site, it has crossed a set of sequence. It has been transcribed, and that sequence is complementary to each other. So see here. So this was a stretch. When it reaches the termination site, already sequence have been transcribed, and that transcribed sequence is complementary to each other. So the complementary base pair, they come together. They form a stem loop structure. The stem loop structure creates a wrenching force on the RNA molecule that is connected to the DNA. It removes the RNA out. That formation of the secondary structure, you call this as a secondary structure. Primary structure is linear. The secondary structure, it forms a stem loop structure. A hairpin structure, it is called. The RNA comes out. So the process is called as, you can see no protein is involved here, no Rho protein is involved here, hence it is called as Rho independent mechanism. Then listen very carefully, I'm not going to dwell deep into this. It is called as a Rho dependent mechanism. Rho dependent mechanism, the Rho is a beautiful protein. It is made up of 1, 2, 3, 4, 5, 6 subunits. It is a hexameric protein. It is in the shape of a spring washer, or it is in the shape of an open door, open donut shape. Okay, it's not closed, it's open. So the speciality of this protein is whenever the polymerase reaches a termination site, in this case, the termination site is called as row utilization site. It is called as row utilization site, RUT. It is called as row utilization site. When it reaches the row utilization site, the row combines with polymerase, forces the polymerase to open up, and the RNA molecule is released. In fact, the way in which the RNA molecule is released out is not fully elucidated, but the row protein influences the polymerase in order to remove the RNA molecule out. Now, very quickly, the DNA molecule has certain region which is ready now for transcription. It has a promoter region to which the polymerase goes and binds. It moves and reaches. As it moves, it adds one nucleotide at a time, and the nucleotides are connected by the phosphodiester bonds, and you have a stretch of single-stranded RNA molecule being produced. When does this production stop? Whenever the polymerase hits a site called as termination site, or it is called as R utilization site. By two mechanisms, the RNA molecule is released out: by row independent mechanism and row dependent mechanism. What is row? The row is a circularly permuted protein, which is made up of 6 subunits and so on. Very quickly, I have surpassed my time, I think so. But anyway, let me continue with the process called as translation. So as indicated in the central dogma, the information present in the DNA molecule is rewritten into another closely resembling chemical moiety called as RNA molecule. Chemical counterpart called as RNA molecule. And from the RNA molecule, it has to be converted to protein. It has to be the information written in 4 letters of nucleotides have to be converted to an information written in 20 nucleotide, 20 letters. The letters are amino acids, basically. Nowadays, it is 21, 22, and so on. Okay, so let's keep it as 20. 20 are unique amino acids, and some modified amino acids are also there. Selenocysteine and so on. Let's not worry about it. So in computer terminology, one information being converted to the other is through an algorithm called as assembler, if I'm correct. Assembler it is called. You have an algorithm which converts the common language into a machine language and vice versa. Likewise, in biological system, you have an assembler which is called as ribosomes. The ribosome is a huge molecule. There are some people who consider even ribosome to be an organelle. It's a huge molecule. The ribosome is made up of 2 components, a larger subunit and a smaller subunit. There is little variation between the eukaryotic ribosome and the prokaryotic ribosome. I'll talk about that in a little while. Okay. So the question is, ribosome okay? The ribosome is the one that rewrites the information written in nucleotides into an information written in amino acids. What is being read by the ribosome is the question. A set of sequences that is read by the ribosome is called as open reading frame. The set of sequences that is read by the ribosome is called as open reading frame. Okay. Does the ribosome reads letter one at a time? No. If it reads letter one at a time, it has only information for 4 amino acids because you have only adenine, thymine, guanine, and cytosine. Whether it is nucleotides, 4 power 2. 16 alone is possible if I'm correct mathematically. So you have 4 nucleotides and the 2 of them, they come together to form words. But you have 20 amino acids. So even a combination of 2 falls short of representing 20 amino acids. Then what about 3? So 3 power 4 is 64. Meaning you have sufficient number to represent 20 amino acids. So basically the ribosome reads these 3 letters in order to find out what is the amino acid that needs to be included in a protein. So a collection of amino acids, you call it as a protein. So the RNA molecule will be read by the ribosome. Since the RNA molecule has some messages in it in the form of ORF, open reading frame, it is called as a messenger RNA. There are some RNA that doesn't get translated into protein at all. Some of them goes into the making of ribosome. They are called as ribosomal RNA or rRNA. Something goes into, takes up a functional role in translation. They do not get translated. They do not convert themselves into, convert into protein. So the information resident in those RNA will not be converted to protein, but they have a function to play in translation. Hence, those molecules are called as transfer RNA because they help in transferring amino acids. Okay, I told you 4 power 3 power 4, it is 64. There are 64 codons. So what is a codon? The 3-letter words in molecular biology, it is called as codon. So basically, the open reading frame has codons in it. So you can see in this case, AAA codes for lysine. See here, K is a single letter word for an amino acid called lysine. A codes for alanine. I codes for isoleucine. And so on. So each of these codons code for an amino acid. How many amino acids are there? 20 amino acids are there. Fine. But 64 codons are there. So what does that mean? Each amino acid is perhaps having multiple words. Like in English, for a particular meaning, you might have synonyms. Likewise, here again, you have synonyms. Most of the amino acid, they have multiple codons. The the feature is called as redundancy. Redundant meaning repetitive. For each amino acid, you have multiple words. Now, like transcription, translation also has the same problem. Where to start and where to end. The translation always starts at a start codon. You can see it starts here, reads one codon at a time and moves on, reaches a stop codon, stop there. So it starts, moves and stops. It initiates, elongates and terminates. Okay, don't worry about the other two pictures in the diagram, which tells you which is not an open reading frame. Do not worry about this. Open reading frame looks like this. You have a start codon. The start codon is usually a methionine. M stands for methionine. In certain instances, it can be valine. Very rarely, it can be valine. But most of the time, it is methionine. So listen very carefully. 64 codons. Among 64 codons, one is a start codon. And 3 of them are stop codons. 3 of them are stop codons. Stop codon meaning it doesn't have any amino acid associated to it. It just plays the purpose of stopping translation. Hence, it is called a stop codon. It stops. So 64 minus 3, 61 codes for amino acid. 64 stops 3 helps in stopping the translation process. So say here, in the case of transcription, the question was, where does transcription starts? We have clearly answered it is at the promoter. Now listen very carefully. Where does the translation start? It starts at the stop codon. But how does it recognize the stop codon? Is an important question to ask. How does it recognize it? All the nucleotide looks similar. Where does it start? How does it know? This is the point where it needs to be started. So you have a special signal which indicates the ribosome. This is the point you need to start translation. If it is translated wrongly, the information is skewed. It produces a wrong information altogether. It has to be started. It has to be started perfectly at the dot on a particular codon, the start codon. How does the ribosome know? Listen very carefully. The mRNA molecule has a particular region which indicates the ribosome that this is the site. You have to bind and you have to start from this codon because it's a start codon. That sequence is called not as a promoter, but as Shine-Dalgarno sequence in prokaryotes and Kozak sequence in eukaryotes. So what you need to understand is there are some special sequences which indicates that this is the point where ribosome has to attach. So I'll give you some details about this. I won't be dwelling deep into it. How does it look like? In the case of transcription, minus 35, minus 10. Likewise, again, here again, it is sequence, basically. But it is an RNA molecule, you know that? So you can see the blue colored region is called as a ribosome binding site, RBS. Then the light green colored region, you know that it is a start codon. Then between the ribosome and the start codon, there should be a small space. The space has to be between 5 to 9 bases. If this is the architecture, the ribosome knows that it is this point at which it has to bind. and starts producing the protein. So this also indicates another information. You can see a ribosome binding site, a start codon, then a stop codon, then a ribosome binding site, a start codon and a stop codon. So you can see multiple information present in the same RNA molecule. We call them as polycistronic. If only one information is present in an mRNA, you call it as monocistronic. If multiple informations for multiple protein, for production of multiple protein is present in the same mRNA molecule, it is called as polycistronic. Now, one more point, it clearly indicates how and where the ribosome binds to the mRNA molecule. Where RBS, you have to have 5 to 9 sequences, then you have to have a start codon. That is the pattern the ribosome looks for. But with which it goes and binds to the sequence. How does it know? How? Imagine very carefully. Like in a Velcro, probably you people will know Velcro. Velcro, you know, nowadays in bags, shoes, and so on, you have a surface with small hooks in it, and you have a surface with tuft of fiber in it. You press them against it, it sticks. So part of the Velcro is in the ribosome and part of the Velcro is in the mRNA. It needs to stick. So with which it sticks is the question. Imagine in your mind, picturize in your mind. You have a ribosome. The ribosome in certain region has another RNA molecule. That RNA molecule is called as 16S rRNA molecule. What is that S? The S is called as Swedberg unit. Swedberg unit. I'll talk a little while later. If you're interested, I'll talk a little bit about it in the later slide. It is called a Swedberg unit. 16S. It is a special type of RNA molecule. The RNA molecule has complementary sequence. Complementary, if there is an A, you have a U there. If it is G, you have a C there, and vice versa. The ribosome has a RNA molecule in it, which has a complementary sequence for Shine-Dalgarno and Kozak, using which it goes and binds. You have a pen, then you have a cap. Click it. Likewise, you have Shine-Dalgarno sequence, Kozak sequence, to which it has 16S rRNA sequence, goes and bind. rRNA, I told you, it's ribosomal RNA molecule. Lakshmi ma'am, have I surpassed the time I have?
Sir, if you wish, you can take for another 5 to 10 minutes, no problem.
Okay, fine. So whatever that I can complete, I'll complete it, ma'am. Because
Okay, completely fine.
Fine, okay. Okay, so translation is nothing but the nucleotide sequence written in small words of 3 letters called as codons is converted to amino acid strings called as proteins. What is required? You have to have a ribosome. What else is required? You have to have Shine-Dalgarno sequence and Kozak sequence present in the RNA molecule. If these two are there, ribosome can identify it. Now listen very carefully. If I'm not able to complete the whole process of translation, no need to worry. Let me complete whatever that is essential and whatever that is interesting. Another component that is very, very important is called as an adapter. Adapter. What is an adapter? Let me give you an analogy. With a caution, don't take it further. For example, you people or all of us would have gone through the problem of connecting an appliance purchased from the Gulf countries, and you try to plug that here in our home. you have a different plug altogether because it is made to connect to a wall socket of 110 volts, if I'm correct. But in our case, it is 240 volts. You have to have a different plug point altogether to which your plug is pinned. So the appliance has an incompatible plug to the plug point that we have. So you know what you do? You buy an adapter. The adapter has to have two things. One is it has to have a surface to which you can plug in the 110 plug, and the other side has a 240 pin using which you can plug into a plug point of 240. On one side, it is the Indian compatible plug pin plug. On the other side, it is an Gulf compatible plug point. So it perfectly connects two incompatible components together. You call that as adapter. Likewise, in RNA molecule, the information is written in the form of nucleotides, 110. On the other side, the amino acids have to be attached, 240. So somebody has to connect it. You need to have an adapter. You have to have an interface. The interface molecule is called as tRNA or transfer RNA. People knew even beforehand, before understanding the structure, understanding the existence of tRNA molecule, people clearly knew that there should be an adapter molecule that connects these two information together. Why? Because these two informations are incompatible. You cannot connect an amino acid directly with a nucleotide. You can't do it. Biochemists and the chemists know that. They are not compatible. They don't have complementary shape. They don't have complementary groups to form bonds. So people were looking for an adapter molecule. Perfectly, they got this adapter molecule. It is called as tRNA molecule. What is tRNA molecule? So you have to expect one surface interacting with nucleotides, other surface interacting with proteins. Like a typical adapter, one interacting with 110, another interacting with 240. That's what you expect here before unraveling the structure of the RNA molecule. The RNA molecule has 3 levels of structure. One is called as a primary structure, secondary structure, and tertiary structure. The primary structure is a single-stranded RNA. If you ask me a question, what is the length of that RNA? Around 73 to 93 bases. Nucleotides, because it's single-stranded, there is no base pairing, it's bases. 73 to 93 bases. It's a single-stranded RNA. Now you know that, as I mentioned in the row-independent termination discussion, there are complementaries. If there are complementary sequences, they come together, they form a stem loop structure. So you can see they form a complex stem loop structure. So the primary structure, by complementarily binding to sequences, they form a secondary structure. So these structures, the stems, you have a 4-stem loop structure or 3-stem loop structure and a stem structure. Okay, so the stems are the regions where the nucleotides have found its complementary bases. The loops are the places where there are no matching, there are no complementarity. Very quickly, let me tell you and also drive home the point of how well tRNA molecule serves as an interface adapter. Listen, this loop is called as a pseudo-uridine loop. Pseudo-uridine loop is nothing but since it has a modified uridine. So you can see this is uridine, and to the nitrogen a ribose is attached. Just take the nitrogen and take the sugar and shift it to a different location. You have pseudo-uridine, a slight modification of the structure. So since this loop has pseudo-uridine in it, you call that a pseudo-uridine loop. So you need to call some loop with a name, is it not? So this is called as a pseudo-uridine loop. In certain instances, you have extra hydrogen. They are called as extra hydrogen added on to the uridine. You call that as dihydrouridine, di meaning two. So since this loop has dihydrouridine in it, you call that as a D loop. Then what about this loop? This loop has beautifully complementary bases to codons. 110 side. You have nucleotide interacting with another nucleotide. So if you have a nucleotide AUG, this has UTC, goes and binds to it. The tRNA can go and bind to it. So this is called as anticodon. So you have a complementary set of 3 letters, which is complementary to the codon. You call them as anticodon. Look at the other side. You have a special structure, which is called as an acceptor arm. What is that acceptor arm? It has a special structure. You can clearly see an extension here. So all the areas are double-stranded. You can see a single-stranded extension here, which is characterized by the presence of these 3 nucleotides, CCA. So it is to this site an amino acid is added. So what does a transfer RNA do? Now you know the answer. You have a codon. The transfer RNA attaches to the codon through an anticodon. I told you 240, 240, 240 pin and 240 plug. On the other side, it has an amino acid. So it plugs, it finds out a particular nucleotides and adds an amino acid. Now the problem is solved. This serves as an interface. I've only talked about the secondary structure. How does the secondary structure form? The bases, they associate with their complementary bases and form a secondary structure. And this secondary structure is called as a clover leaf structure. Clover leaf structure. Since it looks like the clover leaf, or the maple leaf, the Air Canada has that leaf or the Canadian flag has that leaf. Since it looks like that, it's called as a clover leaf structure, or it is called as a tRNA, since it looks like a T. But what you need to understand is this structure is not functional. It has to assume a tertiary structure to become functional. What is the tertiary structure? You can see I'm not dwelling deep into it. It modifies, it rearranges and forms a structure like this. It forms a structure like this. Okay, you know what is happening. This is extended. This angular structure is extended. This angular structure is turned and extended so that the acceptor arm is on one side and the anticodon is the other. Don't you see an L-shaped structure here? So the tRNA, ironically, tRNA is interestingly an lRNA. So you have an L-shaped structure, which is a functional structure. Okay, so The tRNA is also like a bilingual dictionary, because the tRNA is sort of connects the meaning of two words, the nucleotide word on one side and the amino acids word on the other side. It tells the system that for this codon, this is the amino acid that needs to be added. Like a dictionary, like a bilingual dictionary, you know. So you have an English word being translated into Tamil or Malayalam. Likewise, the meaning of a codon is translated into an amino acid. But with the caution, but with the disclaimer that this is not the molecule which is really responsible for maintaining the specificity of the information. Meaning, imagine you have a dictionary with the typos in it. For example, a particular word has been wrongly attached to a particular meaning. Everybody starts using the wrong word for a different context, for a wrong context. Likewise, somebody has to check the quality that for this word, this should be the meaning. A dictionary maker. So the tRNA is not the dictionary maker. The tRNA is just a collection of synonyms. It's just the collection of a word and its meaning. It's just a collection of a word and its meaning. But who has connected this word and its meaning? Probably if we can discuss it later, if I get a chance, or I wish to. Ma'am, Lakshmi ma'am. Lakshmi ma'am, are you there?
Yes, sir. Actually, I was talking, but my mic was on mute.
Okay.
So
because it's I mean, I'm not sure, ma'am, because I've taken more of time. Moreover,
yes,
it's not that there are too much of contents to be covered, you know.
okay. So
Okay.
I think we can stop here. And students, if you have any queries, you can type that in the chat box.
Yeah.
So till now, it's like which topic is important. Those type of queries are there.
Okay.
So I would advise you, if you have any queries for discussion, please do type in the chat box, and this video will be available in our YouTube channel. And we'll be sending you the link of this video once all the sessions are completed.
Okay.
Yes, sure. So I think we can wind up the session right now. So thank you so much, sir, for coming. And I hope our students were able to get a lot more idea about this topic. So thank you so much. And students, you can mail us directly if you have any further queries. We can give you the contact, or we can ask sir to clear those queries also. So thank you so much, everyone, for coming. Thank you, sir. Thank you.
Thank you so much, ma'am. Thank you for the opportunity.
Thank you.
Thank you.